Feeling ready?
Yeah.
Okay. All right. I think we are ready to get started. Sean has passed the first of the snake taming events. It's like getting Zoom to work. So, it's like a pass to us, right?
Yeah. The rest is a cakewalk.
Yeah. I'm excited to introduce Sean Schaefer today. He's defending his Ph.D. dissertation. So Sean came to us in the fall of 2018, all right? 2019, okay, good. Okay, second test. From DePaul College, where he did an undergraduate degree in cell and molecular biology and then a master's in biology. He worked with slime molds, so many of you know that that's like his first and real love, I think. Slime molds. So, he has many others now, but Sean has been an awesome person to have in our lab, and he's my first PhD student, so it's been fun to like come through this with you and learn with you. You'll hear all about his research today, but some of Sean's accolades I should mention is that he was awarded a Department of Energy graduate fellowship, the SCGSR, and so he was able to spend some time out at Lawrence Livermore National Lab where he learned some of the techniques he's using. He also was an awardee of the NASA Space Grant Fellowship, and in addition to those kind of accolades, he's also just like an awesome teammate to have around, really amazing mentor to the other students in the lab, and always willing to lend a hand. And I've really appreciated that. It's been fun. Amazing artist and musician. And so today we're going to get to hear about some of what you've been working on. So take it away.
Awesome. Yeah, thank you.
Oh, I didn't introduce your committee. I apologize. I'm supposed to do that. So your committee, some is in the room, some is online. So Dr. Stuart Grandy, and then online we have Dr. Mark Waldrop, Dr. Caitlin Hicks-Priest, and Dr. Ruth Warner.
Perfect. Thank you. So I'm going to be talking about obviously my dissertation work, what I've been up to the last few years, looking all at plant microbe interactions in the Arctic. So without further ado, let's take a trip to the Arctic. There might be, some misconceptions that the Arctic is like this wasteland or it's always covered in snow. And it actually can be quite a beautiful place. It's full of wildlife. These are different wildlife that I saw the last trip on there. So like grizzly bears and caribou and all sorts of stuff, scenic landscapes. But at the same time, it's a very extreme environment and it can be a pretty harsh place too. So there's pretty low plant diversity relative to temperate and tropical systems. And it's a very short growing season. So anywhere between 45 to 60 days where these plants are really active. But it's almost 24-hour sunlight. So these plants are really active in a relatively short amount of time. And they're very harsh conditions. So very low temperatures in the winter, annual freeze-thaw cycles, acidic pH, and these all kind of contribute to there being not a ton of plants that could withstand these kind of harsh conditions. And one of the unique things about the Arctic is how much carbon is present in the Arctic. So about 50% of the world's soil carbon stocks are in these northern latitudes and roughly 15% of the land area. So it's a very disproportionate amount of carbon in these northern regions. And temperatures are also rising disproportionately. They're rising about 4 times the global average in these northern latitudes. So these are regions that are particularly vulnerable to some of the effects of warming too, which I'll talk about in a little bit. And one of the unique things about the Arctic is the presence of permafrost or perennially frozen soil. So this is a typical soil profile in the tundra in the Arctic, where the top portion is called the active layer. And this is going to be the portion of soil that thaws every spring and summer and then freezes every winter. So it's going to undergo these annual freeze-thaw cycles. And then below that is the permafrost. And that's going to be the soil that's frozen year-round. And this is where most of those carbon stocks are located within these permafrost layers. But as I've alluded to, tundra landscapes are dramatically changing. And this is showcased by permafrost thaw. This warming temperatures are accelerating this. And these are a couple of pictures of what permafrost thaw can look like on the landscape. And this is really just looking at terrestrial. So it's not accounting for like thermokarst lakes or anything like that. But if you look at the top right picture, you can see this massive erosion has happened, really, really dramatic on the landscape. And then these bottom two pictures are different effects that it can have on the vegetation. So sometimes it could increase the growth of one plant, sometimes it could completely remove plants in that vicinity. And why do we care about this? We care about this as ecosystem scientists because there's a lot of carbon in the ground. And just like when you take something out of the freezer and you leave it on the counter, it can start to rot and go bad. It's kind of the same idea here. We have all this carbon that's in permafrost, and when it thaws, it's vulnerable to microbial decomposition. So a lot of that carbon can be decomposed and released into the atmosphere as greenhouse gases. And it could really exacerbate some of these effects of climate change and global warming. And coinciding with this permafrost thaw is this change in Arctic plant communities. So shrubs such as Betula nana, which is a dwarf birch, are increasing in abundance, and they're displacing these tussock sedges in the tundra, process called shrubification. And these images do a really good example of that. So if you look at 1987, it's almost entirely sedge dominated, whereas 2013, it's almost entirely shrub dominated. So a relatively short amount of time, we're seeing this really dramatic shift in vegetation. And these changes in vegetation could have strong implications on the carbon cycle, especially what's happening below ground. And that's really where my work comes into play. I'm interested in trying to understand how these different plant communities are influencing the microbes, which are then changing how much carbon is stored in the soil or released into the atmosphere where it can contribute to climate change. So there's some guiding questions that I've been using to kind of steer this dissertation. The first one, these are intended to be fairly broad too. The first one being is how does permafrost thaw and shrubification influence the below ground microbial ecology of the tundra? So kind of asking what microbes are present and how they're interacting with the environment. And the second question is really building off that first one, is how is shrubification and permafrost thaw influencing the microbial processes? So more specifically, what the microbes are doing, how they're processing carbon and nutrients such as nitrogen and phosphorus in the tundra. And then finally, this kind of ultimate question, which I think I'll get close to answering this, but it's really a loaded question, is will the tundra continue to store more carbon than it releases? And this is really dependent on a variety of things, but we'll take a stab at it too. So to get at these questions, I did a few studies that I'm going to be talking about in a little bit more detail. The first one is a field survey, so it's looking at root-associated microbial communities from different tundra plants. The second one is taking those root-associated communities and putting them onto thawed permafrost and then measuring a bunch of microbial traits and different like substrate affinities of these microbes. And then the third chapter is measuring carbon flows from these plants into the soils and then back into the atmosphere. So it's looking more at like fluxes and biogeochemistry. Okay, so chapter one, this is, again, a field survey where I collected a bunch of roots from Alaska and then analyzed the bacteria and fungi that were growing in tight association with the roots. So the aims of this study, the first one was to compare and characterize rhizosphere communities from these common tundra plants across different glacial mineralogy, so these different sites with different glacial histories. The second aim was to compare the relative drivers between bacteria and fungi, so how much these different host plants and glacial sites will impact the microbial communities. And then the third one was to assess the correlation between rhizosphere and ectomycorrhizal communities in the shrub Betula nana. So first, I'm sure some people are wondering, what is the rhizosphere? The rhizosphere describes the spatial area surrounding the plant roots. And this is soil and communities that are biologically, chemically, and physically distinct from the bulk soil. So the bulk soil being the non-rhizosphere soil. And these tend to be areas with high amounts of microbial biomass and metabolic activity. And one of the reasons for that, plant roots are constantly leaking carbon, kind of feeding the surrounding microbial communities. And this is a theme that I'm going to touch on a few times in the talk. But because of this, we tend to see really active, really robust microbial communities around these root zones. And these tend to be hotspots for biogeochemistry, so especially soil carbon and nitrogen cycling. And that's why it's so important to understand these kind of dynamics. The other piece of background for this study is the ectomycorrhizal fungi. So these are going to be root-associated fungus that forms symbiotic relationship with plants. And they're often thought of as an extension of the plant roots. And this is because they can dramatically increase the root surface area and nutrient uptake. And one of the things about ectomycorrhizae is many of these species can engage in soil organic matter decomposition. So they can interact with the soil in a way that plants that don't have ectomycorrhizae can't do. So this is going to come into play, especially for nutrient acquisition and different strategies by these tundra plants. And along with the plant roots, these ectomycorrhizae make up the mycorrhizosphere. It gets a little bit into semantics. I'll just say the caveat, like when I'm referring to the rhizosphere of ectomycorrhizal plants, I'm kind of like including that with it. So it's the mycorrhizae, the roots, and the microbial communities as well. So our study sites were, samples were collected in the north slope of Alaska near Toolik Field Station. So this is maybe like 100 or 200 miles north of the Arctic Circle. And the sites were chosen based on the differences in the time-sense deglaciation. So this is a strong proxy for soil weathering. The general idea here was that back in the last ice age, this entire landscape was covered with glaciers. These glaciers retreated at different time points. And when the glaciers retreat, the soils are subjected to mineralogical weathering, which means that they can start to get degraded. By choosing these soils with different ages, we're inferring something about the way carbon is either going onto or off of these mineral surfaces. The general idea is that the older these soils are, the more weathered they'll be, potentially the more stable this carbon will be onto the mineral surfaces. So we're using this as like a proxy for differences in the microbial communities. So the hypotheses for this study, the first one is that host plants would explain the most variation in bacterial and fungal rhizosphere communities, followed by their glacial history. So saying that the presence of a plant would outweigh the soil conditions. And then the second hypothesis is that the bacterial and fungal rhizosphere communities would be strongly correlated with ectomycorrhizal fungi. So the communities of ectomycorrhizae would strongly influence the communities of bacteria and fungi in the rhizosphere. So here's the first piece of data I want to show for this study. These are called upset plots, and they're kind of like a complex Venn diagram, only instead of having like 3 or 4 circles, you can have, in this case, up to 6. So this first part is bacteria. The bottom one is fungi. These rows refer to the different plant species, and then these circles, when they're shaded in, are the comparisons that are being made. And the bars represent like the number of bacterial species or the number of fungal species that are within each of these comparisons. So they look a little more complicated than they are. Really, you just need to focus on like the first 6 or so comparisons that are being made. One of the things I found that I wanted to point out, if you look at for bacteria, we see this first comparison. This is going to be bacteria that are present in every single plant rhizosphere. And we see that the proportion of this is very, very high. So we identify the most common comparison are these unified bacteria that are present in every rhizosphere. Then after that, we see these more plant-specific ones. So we see ones for Salix, reticulata, Eriophorum, etc. So we identify this core microbiome bacteria and then the plant-specific ones. For fungi, we kind of see the opposite. So we see that the 5 most abundant comparisons are going to be these fungi that are only found in one host plant. And it's not until the 5th comparison that we identify this core fungal portion of the microbiome. So suggesting that these bacteria and fungal communities are being shaped by slightly different things, where we see that Again, there's this unified bacterial microbiome, whereas fungi might be more host specific. So here's the second piece of data. These are called NMDS plots. They're pretty common in microbial ecology. Really, all you need to know to understand these is that each one of these points represents an entire rhizosphere community. So either the bacterial or the fungal communities. Points closer together are going to be microbial communities that are more similar in composition, whereas points further away are going to be more different. And then each of these facets is referring to the different plants. So these are going to be the bacteria found in Betulina and Petra nigrum, etc. So we can compare between the bacteria and the fungal communities. And one thing I want to point out, if we look at the different colors, we can see that they tend to cluster pretty strongly around the glacial drift. And this is especially true for unglaciated sites. So if we look, it kind of clumps up with unglaciated and then everything else away. And it's really the difference between this unglaciated site and the rest of them that are really driving this variation. So when we put it into a linear model, we can start to look at how these components are explaining the variation in rhizosphere communities. And when we do that, we see that they're quite different for bacteria and fungi. So when we look at bacteria, we can see that 13.3% of the variation can be attributed to the glacial drift, the glacial history of these sites. And that might not seem like a lot, but when you think about environmental data, how many different things can be impacting these microbial communities, 13% can be fairly significant, fairly high explanatory power. And then we see 8.2%, so relatively less explained by the host plant for bacteria, about 9.9 for the interaction effect. When we look at the fungi, we see that only 6.3% of the variation can be explained by glacial drift. So less than half the variation we can attribute to bacterial communities we're seeing in fungi. And we're actually seeing that the interaction between host plants and glacial drift are really the strongest factor in predicting fungal variation. So again, suggesting that the fungi might be more context dependent, might be more host plant specific than the bacteria are. And then the last piece of data I want to show for this chapter is these are called Mantell tests. So what I did is I took a species matrix of our Ectomycorrhizae species. So these are going to be on the x-axis for both of these plots. And then the y-axis, here is the bacterial species. And then the other one is the fungal species. And this is essentially just correlating how correlated these two matrices are. So we see that for bacteria, it's about 30%. For fungi, it's almost 55%. So this is suggesting that the species composition of Ectomycorrhizae are strongly correlated with the bacterial and fungal species in the rhizosphere. And this is suggesting that these Ectomycorrhizae may be shaping the environments and really allowing and disallowing certain bacteria and fungi to thrive in these environments. So the takeaways for this study, the first one is that a large proportion of bacteria are shared across rhizosphere communities, whereas the fungi appear to be more host specific, so a proportionally less amount of fungi are shared. The second one is that rhizosphere bacteria are more strongly influenced by the soil properties and the soil mineralogy than rhizosphere fungi. And finally, ectomycorrhizal fungi are strongly correlated to both bacteria and fungi in the rhizosphere, but in particular, especially rhizosphere fungi. Okay, so here's my second experiment. This is looking at, it's an incubation experiment using quantitative stable isotope probing. So shout out to the folks at Lawrence Livermore for sponsoring me to do the bulk of the methodology of this project, as well as some of the data analysis as well. So again, this is a study where I took thawed permafrost and then inoculated it with different rhizosphere communities. The first aim of this study was to measure taxon-specific growth rates for rhizosphere and permafrost microbes, and this is in the presence with and without exudates. The second aim was to determine whether the intrusion of rhizosphere communities would alter soil organic matter degradation rates, so the decomposition of permafrost. And then finally, to identify microbial communities and microbial taxa that were associated with degrading exudates or soil organic matter. So there's not much extra background to really understand the study, but I did want to point out this aspect here. So as permafrost thaws, it deepens the active layer. And what happens is plants respond to this by growing their roots deeper into the soil profile. And this is to access this thawed permafrost, because this thawed permafrost is relatively nutrient rich. So these plants are kind of incentivized to grow deeper to access these nutrients. And when they do that, they're bringing with their rhizosphere communities. So where these communities collide between these rhizosphere and native permafrost communities, and how those mixed communities will influence the decomposition of permafrost, that's what I'm interested in looking at in this study. So that's kind of where this is focused around. So to accomplish this, we did this in a few different parts. Step one was actually what I did in the last chapter, where I went to Alaska, collected a bunch of roots, and extracted the rhizosphere. I then took those rhizosphere extracts, mixed a couple of them together, and then made that into my rhizosphere inoculant. So I have an Eriophyrum inoculant, which is like the poster tussock sedge. That's what's being displaced by the shrubs. So our shrub being the Betula inoculant. And then I have a permafrost control, and this is going to be permafrost on its own that didn't receive an inoculant. So then step three, I fed these microbes daily. So I either gave them exudates or water. And exudates, if we think back to these plant roots, these plants are constantly leaking carbon, feeding the microbial communities. That's what exudates are essentially. So what I'm trying to do here is simulate the presence of a plant root and feeding in these easily metabolized carbon sources, so like sugars and simple amino acids. So by doing that, we want to see what microbes are responding to this frequent carbon source or the absence of a carbon source. So then step four, at 47 days, I spiked the samples with 13C exudates or 18O water. So these are like an isotope analog. Isotopes are pretty commonly used in ecology for a variety of reasons, but mostly because we can track where it moves from these different biological pools, and in this case, into the microbial DNA. So step five, since we added these isotopes, we're able to measure how much of that isotope gets incorporated into the microbe's DNA. So when microbes are active, they're assimilating these different carbon sources, and the amount of isotope that ends up in the DNA is going to be directly proportional to how fast that microbe's growing. So by having our isotopically enriched samples and our control samples that didn't receive an isotope, we can measure the differences in density and then attribute that to these microbial traits and again, these growth rates. So the hypothesis for this study, the first one is that Betula and Eriophyrum rhizosphere communities would have different rates of decomposition on permafrost. And again, thinking back to the last study, we see there are some key differences in the composition of these Betula and Eriophyrum communities. The roots themselves are very quite different, so it would make sense to think that their microbial communities might be doing something different too. And then the second hypothesis is that soil organic matter degraders and exudate degraders are distinct communities regardless of their rhizosphere. So suggesting that the microbes that are good at degrading exudates are going to be present in both Betula and Eriophyrum, and it won't really matter the rhizosphere context. And then the third hypothesis, that rhizosphere communities would mask and outcompete native permafrost communities. So the presence of a rhizosphere would essentially drown out any native permafrost communities. So here's the first piece of data for this study. This is CO2 respiration over time. So this is a cumulative CO2 respiration plot. And a couple things I want to point out. First, when we look at these two treatments on the top, these are going to be our inoculated treatments that are receiving exudates daily. So no surprise, we see that these are the most CO2 respiration. You can also think of CO2 respiration kind of as an analog for how active the microbial communities are and how metabolically active they are. So we see that the rhizosphere inoculants that are receiving exudates are really high respiration rates, followed by the rhizosphere communities that are not receiving exudates. So these are somewhere in the middle. What's interesting here is when we look at the permafrost groups, we see that whether they're receiving exudates or not, it's really not changing their growth rates. And it was kind of a surprise because we're thinking that if we're adding in this very metabolically available carbon source, we should expect to see more respiration coming from these permafrost communities. And there's, a couple reasons why this could be the case. It might just be like the metabolic profile of the communities. We might have fairly low microbial biomass in uninoculated permafrost, but nevertheless, it's quite unusual to see that. But what's really cool about QSIP is that we can get really high resolution data, whereas with traditional stable isotope probing, you kind of get like a community aggregated response. With QSIP, it combines stable isotope probing and DNA sequencing. So we get growth rates on the taxon level or like the bacterial species level rather than the entire community. And that's what this is showing here. So our x-axis is the log growth rates. The further shifted to the right it is, the faster these microbial taxa are growing. And then we have our different plants. So again, we have Betula, or the shrub, our sedge, and then our uninoculated permafrost. And one of the things I want to point out here is that when we look at Betula, we see a whole ton of points that are just stacked on top of each other. We don't see this separation of groups. So these groups are going to refer to the isotope treatments, and they're represented by different shapes. When we look at Eriophorum, we see all these diamonds that are shifted to the right. And these diamonds are going to be our treatment that didn't receive exudates. So we're seeing that all of these microbial communities in Eriophorum are growing really, really fast when there's no exudates present. And we don't really see as many with exudates present. And when we stack them all on one plot, then we start to see this story in a slightly different way. So we see this whole large cluster of Eriophorum bacteria that are growing really fast without exudates. And then we see kind of this stack of Betula followed by permafrost and Eriophorum that are receiving exudates. So we're kind of seeing this like hierarchy of growth rates. And what's really neat is that we can start to plot these different treatments against each other, and we start to reveal different microbial strategies and even like different plant strategies. So here we have the x-axis is our growth rates with no exudates, and then the y-axis is going to be growth rates with exudates. Each point on here is referring to a bacterial species. So these bacteria here on the y-axis, these are going to be taxa that are only found when exudates are added. So these are going to be our exudate specialists. Whereas on the x-axis, we're seeing the taxa that are only present when exudates aren't added. So these are likely soil organic matter specialists. And then this one-to-one line is kind of like an equivalence point. So these are going to be the taxa that are more or less growing at the same rate when there is exudates or when there isn't exudates. So this is suggesting that there's not a strong preference toward exudates or soil organic matter. But what's really neat here is when we look at this cluster over here, again, we see that all of these belong to Eriophorum. So we see that these Eriophorum plants are harboring a bunch of bacteria that are really good at growing fast when there's no exudates present. And this, again, this is suggesting that they have to be getting their carbon from some source. That carbon is likely present in the permafrost. So it's likely that they're going after soil organic matter. Whereas when we look at Betula in our uninoculated permafrost, we could see there's more of a mix between exudate degraders and soil organic matter specialists. So we can look at this data in a slightly different way. And here I'm showing the average growth rates up top So these shapes are really referring to the kind of treatments that I was just talking about. So we have exudate degraders. We have the microbes that don't really have a preference. They're stimulated by exudates, but they could be degrading exudates. They could be degrading soil organic matter. And then we have our soil organic matter degraders. When we look at Betula, we see that the growth rates are relatively even across the treatments. And the same can be said for permafrost, albeit at a slightly lower growth rate overall. When we look at Areophrum, again, we see that this soil organic matter degraders, the growth rates on that is they're growing at a much higher rate than in the other two treatments. So when we sum up all these growth rates, it tells the same story, but like in a more exaggerated way, right? So we look across the sum of all of these growth rates for Betula, we see that they're relatively even across the groups. There's like relatively as many soil organic matter degraders as there is exudate degraders. However, with Areophrum, we see it's almost entirely dominated by soil organic matter degraders. So there's much less bacteria that are associated with root exudates. And again, with uninoculated permafrost, we see it's relatively even, but albeit like much lower growth overall. So the takeaways for this study, the first one is that Betula and Areophrum displayed similar respiration rates with strikingly different substrate preferences. So the Areophrum appears to be strongly associated with soil organic matter degradation. It's kind of interesting because the total output of CO2 is more or less the same between these groups, but they're clearly going after different carbon sources to do that. The second takeaway is that uninoculated permafrost had low microbial activity, and this is suggesting that rhizosphere communities drive much of the biogeochemistry in thawed permafrost systems. So permafrost on its own may be relatively less active. And then finally, that shrub encroachment is likely to alter microbial community dynamics, potentially decreasing the prevalence of soil organic matter targeting microbes that are associated with Areophrum plants. So the replacement of Areophrum with Betula may decrease soil organic matter degraders. Okay, I think we're good on time. The last chapter of my dissertation is looking at, say, mesocosm and greenhouse incubation, again using stable isotopes, but this time in a slightly different way. The aims of this study was to determine soil organic matter respiration rates associated with different plant types, different permafrost types, and elevated CO2 levels. And we also wanted to assess how shrubification and permafrost thaw would impact the amount of decomposition of permafrost. And then finally, to disentangle these plant microbe permafrost interactions. So a quick review on the carbon cycle in the tundra. It's really similar to carbon cycles in temperate systems. However, we have the presence of permafrost. We have this pool of atmospheric CO2 through photosynthesis that gets incorporated into plant biomass. And then, as I've mentioned, plants are constantly leaking carbon, roots are dying off. This forms these rhizodeposits, and a portion of that will form into soil organic matter. And this is going to be the carbon-rich, the organic portion of the soil. A proportion of the soil organic matter is then decomposed. This is a source of food for some microbes. So some of this is decomposed and released back into the atmosphere as CO2. The difference here with the tundra is that we have this presence of permafrost. When permafrost thaws, it essentially enters into the soil organic matter pool. So the organic parts of the permafrost can then be degraded by microbes. So what we did here is we're using atmospheric 13 CO2. So again, using this carbon isotope, and we're doing that because we can track it as it goes into the atmosphere, into plants, soil organic matter, and then eventually into, or what's released back from soil organic matter into the atmosphere. And this question mark is really where this study hones in on, right? Like, we have a better idea of how much carbon goes into the vegetation through photosynthesis, and we could use that to kind of calculate how much would be of the biomass is formed into soil organic matter. However, the big unknown here is how these plants are impacting the amount of soil organic matter in of thawed permafrost that's decomposed and put back into the atmosphere. So the other piece of background I want to explain really quick is this concept of priming. Priming is the excess decomposition of soil organic matter stimulated by easily metabolized carbon input. So as I keep mentioning, plants are constantly leaking carbon. They're feeding the microbial communities, and they're doing this because they want to stimulate the microbes to start breaking down soil organic matter. Plants can't do this on their own, but they need the nutrients that are locked in the soil organic matter. So they use these microbes to increase this decomposition so then they can take up the nutrients that are within these soil organic matter complexes. And what happens is that you can have more carbon leaving the system and leaving the soil than what's going in. And that's represented by this purple arrow, where we see more carbon is lost from the soil than what the plant's putting in. And that's what we wanted to measure here. So to do this, we used biogeochemistry with an isotope labeling cabinet. This is like the bane of my existence for like 3 years. When I came here, I essentially like inherited a box with some grow lights, and it many things to test, and a bunch of holes to drill, and all these different sensors and components to measure. You know, shout out to Nate Alexander and Lucas Bernhardt for really helping spearhead this and figure all this stuff out. And the main goal of this was that we wanted to measure soil respiration. So I'm not going to go into too much detail on how we parsed through all these different sources. What I did want to mention here, though, this R total value is essentially what I was measuring on our gas analyzer. But what we wanted to know is how much of the CO2 is coming from permafrost and how much of it is coming from plants. And by using a 13 CO2 isotope, we can distinguish that, as well as using these plant and no plant controls. So using a lot of whiteboards and algebra and isotope mixing models, we were able to parse out these different sources of CO2. The design of the study is relatively simple. It's a 3x2 by 2 factorial design. So we're testing for the effect of plants. So we have, again, Betula, which is our poster shrub. We have Eriophorum, which is our poster sedge that's being displaced by the shrub. And then we have our no plant controls. We have 2 permafrost types, so these are going to differ in the age. We chose the end members, our super young permafrost at Achillic 2 and our super old permafrost at our unglaciated site. And then we also have 2 different sides of the chamber. So these are going to be elevated CO2, so our 700 parts per million CO2, as well as ambient 400 parts per million CO2. And again, we want to see how these factors influence the overall respiration as well as priming of soil organic matter. So the hypotheses for this study, the first one was that Eriophorum would have a higher priming effect than Betula due to the lack of ectomycorrhizal associations and reliance on microbial communities for nutrient acquisition. So as I referred to earlier, ectomycorrhizae can engage in some level of soil organic matter decomposition. Because Betula has those, they'd be like less incentivized to feed the surrounding microbes, whereas Eriophorum does not have mycorrhizae. They're entirely reliant on rhizosphere communities for this kind of nutrient turnover. Second hypothesis is that recently glaciated permafrost would have a higher priming effect than our unglaciated permafrost due to less mineral protection, greater desorption abilities. Essentially, what I mean by that is the carbon is going to be more available in less weathered sites. So the microbes will have an easier access to that, hypothesizing that would also result in a higher priming effect. And then finally, the last hypothesis that elevated CO2 levels would produce higher rates of priming due to increased photosynthetic abilities and root exudation. There's been tons of studies that show plants grow bigger with elevated CO2 levels. The idea here is that they're growing bigger, they're able to fix more carbon, more of that's going into the ground, more priming is going to occur. So here's the first piece of data for this study. This is looking at total respiration rates. So this is both the plant-derived respiration and permafrost-derived respiration. And we can make a number of different comparisons from this. So These shapes are referring to the different permafrost. So again, we have achilic 2 is our super young permafrost and the super old permafrost. The 6 on the left are going to be at ambient CO2 levels, whereas the 6 on the right are elevated CO2 levels. When we look at the Betula groups, we see a consistent pattern where our achilic 2 young permafrost is going to be less total respiration, whereas our unglaciated site, we see relatively more respiration from those groups. And we actually see the same thing for our Areophrum treatments in ambient levels. Where it gets weird is when we look at our Areophrum elevated CO2 levels, not only are we seeing the opposite trend, but we're seeing a very exaggerated version of that. So we're seeing really high respiration in this young achilic 2 permafrost with Areophrum, whereas relatively no respiration. And in this case, it's even less than our no plant controls in this unglaciated elevated Areophrum. But where it gets even more interesting is where we see these sources of CO2. So when we partition out how much of this CO2 is coming from plant-derived versus coming from permafrost-derived, it tells a slightly different story. So if we look at our Betula treatments, we see that in every instance, there's more plant-derived carbon than there is permafrost-derived carbon. And in some cases, it's like quite a bit more, right? Especially like for our unglaciated, it's like almost entirely plant carbon. Whereas when we look at for Areophrum, we see the opposite. We see that most of our carbon is coming from permafrost-derived respiration. And even in this group with the achilic 2, where we see this really high respiration rate, we're still seeing that there's a higher proportion of that carbon coming from permafrost instead of plant-derived respiration. And this is suggesting that Areophrum, again, has these microbial communities that are really good at degrading soil organic matter, whereas Betula appears to have more recycling of this newly fixed plant carbon. And then the last piece of data I want to show is looking at how these kind of varied throughout the experiment. So our y-axis is going to be our priming rate, the colors representing the plant types and the shapes of the permafrost. Early in the experiment, we see that Betula had a stronger priming effect than Areophrum and our no plant controls. And at about 2 weeks or 3 weeks into the experiment, we start to see that shift. So in most of the Betula groups, we see that there's a less of a priming effect than our no plant controls, as well as Areophrum, other than this one treatment that's seen an increase. And then about 3 weeks in or so, we see this kind of shift. So we see that Areophrum was much more active later in the experiment, whereas Betula started to taper off. And I wanted to show this to give a sense for how context dependent these systems are and how variable, not only between the groups, but even within a group. Like some of these error bars are gigantic. And that's really just showing, again, like how context dependent and how variable these systems are. So then if we look back, if we take what we learned from this experiment and put it into our conceptual diagram of the carbon cycle, we can start to get a sense for like what's happening with this carbon. So if we look at sedges, we see some of this plant carbon is being recycled, but we see more of it is actually coming from soil organic matter. So we're seeing more of the soil carbon lost there. With shrubs, we see the same idea, although like the pool of plant carbon might be bigger. So this recycling of plant carbon appears to be a larger flux, and then comparably less carbon being lost from soil organic matter. So the takeaways for this study, the first one is that plant species, permafrost type, and atmospheric CO2 levels significantly influence soil priming rates and carbon cycling in thawed permafrost. And this interaction, again, is very highly context dependent. The second takeaway is that plants show strong temporal dynamics, suggesting that they may affect priming at different points of the growing season. And this is something that we've been thinking about as the group and really trying to figure out like what a great next step for a study would be. And this idea that Betula may be more active earlier in the season, their roots are comparatively much more shallow, presumably would be thawed earlier in the season, they may be more active, whereas Areophrum tends to have deeper roots. So it's a concept that we may be exploring. And then the final takeaway is that Betula is more associated with plant-derived carbon, whereas Areophrum is more associated with permafrost derived carbon. So we see this in this study, and then we also see this in the second chapter looking at the microbial physiology and traits. Okay, so putting it all back together, zooming out a little bit, and like what does this mean for for Arctic carbon cycles and global carbon cycles? The first one is that shrubification will alter microbial community compositions and the below ground ecology. So the first chapter, there's a lot of shared bacteria between these plants, but there's also quite a bit of variation. So more shrubs coming in, replacing tussocks is going to change the microbial communities. The second thought is that this increased betula growth at the expense of Eriophorum may mitigate carbon losses by decreasing soil organic matter targeting bacteria. And I have to throw the caveat that this is really just thinking about the microbial community traits and dynamics. So there's a lot more that goes into this, especially like how betula can influence soil temperatures and like albedo effects. So I'm not really thinking about that when I say this, but really thinking just from a microbial lens, it has this potential. The third thought is that permafrost mineralogy and its interaction with plants and microbes ultimately will determine the magnitude of priming effect. So like the maximum amount of priming is very much determined by these plant permafrost interactions, kind of setting the stage. And then the final thought, whether the Arctic remains a carbon sink is ultimately determined by the amount of permafrost thaw. So when permafrost thaws, much of that carbon is going to be decomposed and released to the atmosphere. The shift in shrubs may mitigate some of those losses and keep more in the ground than if there wasn't shrubs, but ultimately, the extent that the tundra will be this soil carbon preservation is really dependent on how much permafrost is going to thaw. So with that, I'd like to thank all my friends and family, funding sources, the folks over at Tulik, my committee members, my collaborators, and then end with a slideshow of all the fun things I did over these 5+ years, both in the lab and in the field, and then also out. So with that, I'd like to open the floor for any questions.
But maybe that would say there's more about
Yeah, Danny.
That was awesome, Sean. Congratulations.
Thank you.
Really, really cool work. Okay, so you had this the first bullet point of your last slide was that shrubification will alter microbial ecology within Arctic. Does your work or does anybody else's work sort of put that on its head of like how microbial ecology is affecting the rate of shrubification? Or is it really just driven by like air temperature? Like, what's the feedback there?
Oh, that's a good question. So the question was more or less looking at my last slide was looking at shrubification and how that's changing the microbial ecology. The question was, is the reverse true? Is the microbial ecology kind of dictating what plants are able to live there? And I would imagine it's probably a good mix of both. That's a really cool lens to look at it. Certainly like the presence of the plant friends, the microbial friends will allow certain plants to have like an advantage in certain environments. I think that might speak to more to like the mineralogical dynamics. So we see like there's pretty big differences in the microbial communities based on the soil types and like the properties of the soil. So certainly that I think that kind of plays into the interaction effect. So like the context of the soil and the microbes present there will then influence what plants will thrive and do well. Yeah. Yeah, Mel.
So I'm curious, if shrubification might mitigate some carbon losses, do you see further successional changes? And what might that be, and what would your thoughts be on more or less mitigation of carbon?
Oh, that was a good question. So I'll repeat it for Zoom. The question was, Strubification, having this potential to reduce carbon losses, do I see a further successional, kind of like the next like plant succession of that? And in some areas, I think there's probably going to be an expansion of the boreal forest. So there may be like the black spruce trees growing further and further north. So I guess if there was a next succession, it would be the presence of even more like larger shrubs and more trees. So we're seeing not only like the increased prevalence of shrubs, but also like greater biomass in the ones that are already there. So I think the next succession will probably be more plant associated biomass. And it's kind of like the jury's still out for like exactly how this the presence of the shrubs will affect like carbon balance long term. Hopefully it's going to store more carbon, right? And that's kind of the hope. If it can mitigate some of the below ground effects while storing more carbon in the biomass, then that'd be an excellent next step. Yeah, Nate.
Right, I've been threatening this question for years for Sean, dissertation question. I'm really interested in chapter 2 in the area from like associated SOM degraders and how they don't seem to care for the exudates. I'm wondering, I guess, why you think, or slash how you think Areophyrum is kind of selecting its, helping to shape its microbiome, especially if they're not reacting to those like simple carbon substrates we associate with root exits and like associate with priming traditionally. I guess it's like they are priming, but not the way I think about it, through like stimulating microbial activity with those exits. So I'm just curious what you think, how they're shaping their rhizosphere community.
Yeah, that's a great question. Nate asked essentially why does Areophyrum have these soil organic matter degraders? And when we think of priming, we think of plants releasing these easily metabolized sugar compounds, and we see that the Areophyrum communities are less reactive to that. And it went against our hypothesis, right? Like I thought that because Areophyrum doesn't have these ectomycorrhizae, they probably have communities that are really responsive to root exits. And we saw the opposite. And I think why that is because the way Areophyrum roots grow, they grow essentially in like a ball of dead root biomass. So Areophyrum releases like it grows like 3 or 4 new roots every year, and then those roots just slowly like decay and die off. So the roots are surrounded by a great deal of necromass and soil organic matter. And I think that's what's reflective of these communities, is that we are seeing the proportion of like soil organic matter and root necromass is so much higher than root exits that it's just like completely flooded out by these soil organic matter degraders. Yeah, Elza.
I have another plant question. From your first chapter, I was like looking at the NMDS plots, I noticed that it seemed just like from my eye, like the Salix and Betula almost had more grouping than the other plants. And I was wondering if you noticed that, and if you have any thought on like how plant functional type or like this type of plant might influence like the rhizosphere.
Oh, that's a good question. Yeah, Elza asked how like the microbial community seem to have more groupings with Betula and Salix plants, and if I had any thoughts why that would be the case. It's interesting that you noticed that. I did notice that a little bit. I haven't really poured through that data in a bit. I should revisit it, but it's interesting that you point that out because those are the ectomycorrhizal symbiotes. So those are the shrubs. It might be speaking to that these deciduous shrubs might have these more kind of distinct nuances in the communities. And that might also be speaking to this this link between ectomycorrhizae and rhizosphere communities, whereas the other plants that I looked at don't have that same association. So we could be seeing like further specialization within these plant functional types based on the species of ectomycorrhizae that are present in the roots. So that'd be my hypothesis. That'd be interesting to follow up on. Yeah.
Up there, is there anything on Zoom that we need to be paying attention to?
No questions on Zoom so far.
I'm trying to look at those. Let's all thank Sean. And then maybe you want to let your committee know that we're gonna just need to change rooms and then.
Yes. Committee members, we will change rooms and be back online shortly. Thanks everyone for looking online. I really appreciate it.
And changing price for cookies and elevated ambient CO2 beverages. Of course, it's